Welcome everyone, my name is Carola Lopez and this is our poster presentation for Bio 440 Functional Genomics with Dr. Butero. Our title is Evaluating Paramecium Database, Strength Limitation and Functional Genomics Applications. Many community-based databases exist to store genomic information for organisms used in genetic research. We outline the key features an affecting genome database should provide, and we will be evaluating the Paramecium database to see if they meet this criteria.
Hello, my name is Andrew Ferris, and I'll be going over the methodology section of our poster. For the methods, we tested the main tools the Paramecium database offers to see how well it works as a genome database. First, we ran blast searches in both databases and compared the outputs, how easy they were to read, and how adjustable the settings were. We then checked the alignment tools to see how clearly each one showed sequence similarity. We also tested how reliable the search and retrieval tools were by looking up genes and checking how much information each database provided. After that, we explored the genome browsers to see how easy they were to navigate and how much detail they gave. We reviewed the gene annotation pages as well, looking at how complete and helpful the information was. Finally, we checked how well each database allowed us to export data in their standard formats. Overall, these steps helped us see whether Paramecium database meets the basic expectations for a functional genomics resource.
Hello, my name is Lee Stickling, and I'll be going over the results, beginning with the BLAST results. Paramecium database will blast a nucleotide sequence put into it against the genomes that are included on the database, and then it will show your BLAST results in a tabular format. You do have to click into each individual tab to see the hit results for that specific genome. It has some pretty good tools like the query coverage, lets you visualize which parts of the gene input is being covered by the hit result. The genome browsers, as compared to NCBI, Paramecium database's genome browser was pretty bare bones. It does have gene annotations, though not nearly as many as NCBI has, and it does allow you to do things like custom tracks. One neat feature that it had was, again, a good visualization tool, which is the GC content, which is this line here. So you can kind of see at a glance, without having to zoom in on the gene, an idea of what the sequences in that region look like. For annotation, when you do click on these individual genes, you can get pretty detailed results for the genes on Paramecium database, and they're more curated than you might see on NCBI for things like whole genome duplication research and comparative genomics and stuff like that. Alignment tools, the Paramecium database's alignment tool was basically just the same as doing a blast, but it did allow you some visualization. However, the refinement options just weren't there. You couldn't choose an algorithm or anything like that as compared to NCBI's alignment tool, which does allow you to do so. Other than that, the exportability and format of the data from Paramecium database, you did get the basic standard formats. You can get FASTA for your blasts and CSV reports for your blasts, as well as FASTA sequences for like genes and stuff like that. Sequence search and retrieval, they do have a tool called Sherlock, which allows you to look at individual genes, though it does lack the non-coding genes. Usability was pretty lax. Paramecium database was slow and clunky in many areas and a little bit difficult to figure out as compared to NCBI. And then the metadata and documentation, each page did have references, and you could get metadata on genes and the version numbers of different tools and annotations. But the ParaWiki page featured on the website did look like it was unfinished and did feature a good number of dead links.
Hi, my name is Lexi, and I will be sharing our overall conclusions of the Paramecium database. Through our project, we found that the database meets the minimum requirements for an effective database, but lacks visualization and other resources that allow accurate and useful comparisons. Some of the barriers Lee mentioned within the BLAST tool really limited our ability to compare results. The tabular format made comparing and analyzing data very difficult, in contrast to the comprehensive lists generated by NCBI. The lack of internal links and ability to refine search parameters posed many difficulties in analyzing the data, and overall, we just found it not to be very user-friendly. Despite some of these setbacks, ParameciumDB does offer strengths for researchers focused on species-specific genomic features. Some of these features are listed within gene pages and browsers, such as expression profiles, homology data, functional annotations, and more. The goal of the project was to identify if Paramecium database met the key requirements for an effective genome database. We conclude that the manual curation of PDB annotations, gene profiles, and addition of mitochondrial genomes make it a very great resource to enrich studies for gene-explicit studies. However, it is limited in its performance and efficiency in terms of species identification studies, where researchers may be more successful using a comprehensive database like NCBI. Thank you.